would like to introduce our guest speaker, Joel Williams, who is over here. So Joel is an independent plant and soil health consultant, a healthy soils advocate, and an educator on soil biology, plant nutrition, and agroecological farming systems. He has a keen interest in managing plant diversity, soil microbial ecology, and plant and soil nutrition. He has lectured to farming audiences internationally and works extensively throughout Australia, Europe, and Canada, which is where his home base is. And I had the pleasure of hearing Joel speak last year at the RCS conference, and he's a very engaging and knowledgeable speaker, so I'm sure you're all going to get a lot out of today. So over to you, Joel.
Pleasure to be here. Thank you very much, John, for that introduction, and thank you all for coming along. Really looking forward today. What a beautiful part of the world. I've never travelled much in this particular region of SA, so had a beautiful drive through the valley here this morning. Very, very lovely. So pleasure to be here. I was just over on Kangaroo Island, giving a talk on Monday, and that was also very, very nice part of the world. You live in generally a very nice area here, so pleasure to be here. And yeah, let's dive into it, because I've got lots to get through. I usually have a bad habit of preparing a few too many slides, and I'll have to keep my eye on the time and make sure we get through everything. But if I could just get a little feel for kind of who's in the room today, just to maybe help me steer some examples and that kind of thing. Are we mostly a grazing kind of, or who's maybe just livestock? Who here would be purely livestock grazing operations? Okay, so that's what I was told, that it was mostly going to be that. And so is anybody mixed, livestock and kind of cropping? And do we have any pure cropping? Okay. Any horticulture or vines or anything? Okay, yeah. Okay, couple. Did I miss anything else? Maybe some other government people and things? Forestry. Okay. Wow. Awesome. I don't think I have too many examples from forestry, but I'm sure some of the general soil health things will be information will still be relevant. Yeah, I'm sure. Okay. All right. So for the first session, we're going to talk on plant diversity, and really, I'm going to take that through a bit of a lens of soils, just thinking about plant diversity, how that can change soil properties, and we'll look at a few examples of that. And then we'll kind of also just talk a little bit about, I'll do a little bit on the crops here, particularly just a few interesting examples which lead us into the more diverse discussions. But actually there's plenty of very fascinating examples happening even as we step from 1 to 2 plant species, and we just have 2 root systems interacting, some changes that occur even at the level there, and then we'll move on to some more diverse mixtures as well. So we'll kind of just take a bit of a broad look at this, and also, of course, consider a little bit about soil carbon, organic matter, but I think we'll talk a bit more about that when we go for our walk later this afternoon. And then, yeah, we'll also do another presentation after our morning tea break as well. But we'll start off with diversity for now, and let's dive into it. The first thing kind of to talk about here is this. When we start thinking about soils, this idea, this kind of well-known quote, some of you might have heard this one, this idea of, feed your soil and it will feed you. I really quite like this quote. I've been a fan of this one for many years. I have a fridge magnet of it at home. And it's kind of commonly used, particularly in the organic world. You often hear this one. And, of course, it conjures up these thoughts of this kind of concept around, soil care, soil nurture. We need to improve the soil. We need to feed the soil. We need to make the soil healthy. We need to work on that soil. Let's improve soil health. And when we make the soil healthy, then the plants that grow in that soil will also be healthy. That's the kind of the idea. So it has this kind of bottom-up approach. It says, well, let's try and align the soil, adjust the pH, let's try and build organic matter, let's apply fertilizer, let's apply organic amendments, let's compost, let's feed, let's change the soil, let's improve that soil health, and from that healthy plants will grow, healthy animals, healthy humans, all that kind of thing. So it's very much this bottom-up view. It's kind of very focused on improving the soil for those gains, and then within that, we could then kind of break that down into, okay, what do we mean when we talk about soil health? Things like the chemical properties, the physical, the biological properties, and we could kind of work through these and kind of think about it in this context. Well, I'm trying to improve the pH, I'm trying to add the nutrition, add fertilizers, for example. I'm trying to build the biology. I'll use things like compost or manures to feed my organisms, feed the life in the soil, or things like, to improve the structure, maybe a mechanical intervention, subsoiling, again, maybe applying compost to improve the organic matter, to improve the structure, these types of things. So we could go through all these types of things, and we consider this concept of soil health in the context of, changing some of these parameters, changing these variables, trying to improve that soil. And it's, of course, what we see now with this, generally with all of this discussion around things like regenerative agriculture, we have a slightly different view of soil health emerging, what we might call the top-down view, captured perfectly in this quote here, soil without biology is just geology. Acknowledging that actually the very process of soil formation is driven by plants, it's driven by life, driven by biology, driven by organisms who grow on rock, on parent material, and over time the interaction of the living organisms, the biology onto that rock, over time of course helps to form soil. So we see that in this kind of ecological succession type point where we have, tectonic upheaval, volcanic activity, this type of thing, rock coming to the surface. And then we have all of this kind of microorganisms growing in there, the mosses, the algae, the lichens, the bacteria, these types of things. Small things will start to grow on there. What are they doing? They're stripping some of the minerals out of the rock, the inorganic nutrients out of that rock, growing their bodies. And then their bodies grow and die, grow and die, grow and die over many generations, over many thousands of years. And so, of course, they are depositing their dead bodies into that rock. So we have an organic fraction now, organic material, coming into, mixing in with the rock, which is where beginnings of soil formation, pockets of soil formation. This happens over many thousands of years to a point where, of course, some now higher level plants can move in. And typically we're talking about our pioneer type plants, those early successional plants that can move in, and then they start growing and dying, growing and dying, growing and dying. And again, they're depositing their dead bodies, building up that organic horizon again, a little more, accelerating that soil formation process. And onwards we go. We switch at some point to more perennial-based plants. Perennials, they allocate more carbon down below to their roots, being that they are perennial, meaning they'll come back next year, so they invest more heavily in roots as compared to our annuals. So as we shift into perennials specifically, we get even more carbon accumulation down below ground in that soil, really accelerating this process then of soil formation, and onwards to shrubs and forests and all of that kind of discussion. So point being is that without the action of that biology, that life, and the examples of course would be the moon or Mars, we have the geology, we have the rock there, but we don't have the life there. We don't have the interacting organisms driving that process of soil formation. And so if we think about in that context of top-down, of soil formation, we could just extend that thinking to the concept of soil health. And I know, depending, the concept of soil health is also quite a broad topic, a little fuzzy, somewhat mixed in its various definitions, but nonetheless, this is the essence of the idea of why do we grow cover crops? The idea of a cover crop is, well, I'm going to grow a plant to improve the soil, to change the soil. I'm going to grow a legume to bring nitrogen in. I'm going to grow buckwheat to scavenge some phosphorus. You're growing a plant to improve the soil. So it is this idea that says, okay, with the action of that plant, of photosynthesis, of the interacting organisms, as they grow in that soil, they can also change the soil. So we have this then top-down view, and I think both views are fine. They are complementary. It's a bit of a two-way street. Of course, the health of the soil changes how plants grow, but of course we can choose strategic plants to also then work down and change the soil equally. So it's a bit of a two-way street. So I think it's a different view of the same thing, but I think there's a lot of course, traction and interest in this top-down view now, acknowledging that, okay, we can also use plants specific plants or diverse mixtures of plants to also help to improve soil or heal the landscape, this type of idea. So I think it's a good shift in our perspective happening these days. Now, key to this, however, when I say that plants can improve the soil health, change the soil health, I am really predominantly talking about roots. It's the action and the activity of roots that grow into that soil that really are the key direct drivers of that change. Now, of course, roots and shoots are connected. They are interrelated. They are the one thing. They are the one plant. And if we have good, healthy biomass above ground, well, sure, generally we're going to have good, healthy biomass below ground. There is a relationship there. But in terms of more of the direct benefits to soil structure, to aggregating, improving porosity, to nutrient cycling, to depositing those organic materials and building carbon, etc. In terms of all of those benefits to the soil, it's really the roots that are the primary drivers, direct drivers of this. And so this is important in our mindsets, that when we go and look at the landscape and assess of soil health or look in a field, of course, what's happening above ground is very important, but it's key that we also consider what's really happening below ground. So, of course, like we all do these days, take our spade with us. Take our spade with us and make sure we assess the roots. What are the roots looking like? Let's not just judge the success of a cover crop or a pasture solely by how it's looking above ground. Let's make sure we have a bit of a dig and explore some of the different root interactions and root biomass, root depth, root architecture, these types of things of some of our below ground, of some of our plants at the below ground. And so that's the kind of the key message. And really, that's that broad question then when we say, well, how can we improve soil health? If I want to improve that soil health, how do I use plants? How do we improve things? It really is key that it is about roots and living roots. And we'll talk a little bit more about this. I think when we go outside, we'll have a little bit of a discussion around some of the slight nuance and differences between roots and the living roots. By that I mean root litter, root residues that are dying and decomposing versus living roots that are releasing those root exudates. We'll have a little bit of a discussion about that out in the field. So roots are the key, and therefore the next extension says, okay, well, what plants do we then use? And that really depends on your goal. What plants do you want to help heal the system? What plants do we want in our pasture? It depends on what your goals are, as to which maybe is the most appropriate species for that. Your local area, your conditions, these types of things. Are we looking for nitrogen? Are we looking for phosphorus release? Are we trying to capture nutrients and keep them in that biomass of the plant and prevent them from leaching through, for example? Are we just trying to cycle nutrients, make them available, unlock them, release them for plant growth? In a cropping situation, we might be particularly interested in some of the disease suppressive properties of brassicas, for example, some of that biofumigation effects that they can bring. Or are we trying to support the ecosystem in a broader landscape level way, using pollinators, flowering plants, attracting in beneficial insects, creating habitat for the ecosystem, for the wildlife? Are we trying to suppress weeds? Again, maybe more relevant particularly relevant in a cropping situation, and I'll acknowledge, of course, if the goal is weed suppression, sure, that is where that above ground biomass is absolutely very important. We want that shading effect. We want to suppress the weeds. Well, what's happening above ground is very important from a weed point of view, for sure. Are we trying to increase soil organic matter? And we'll talk about that. That means you definitely want to grow more roots, deeper roots. And we'll talk about that, I think, when we go outside. And same with compaction. If we want to alleviate compaction, improve soil structure, we have to get deep roots down into that soil to restructure that soil using those roots to help us with that. And so these are some specific examples of what we might be trying to achieve. But generally, most of you are probably going to be interested in quite a few things on that list. There's probably not just one thing there that you're interested in trying to improve in your system. It's probably multiple. And that, therefore, leads us into this discussion about diversity. If we want multiple functions, if we're looking for multiple benefits, well, it's likely that a multi-species mix is going to provide some of that multifunctionality. And that's the essence of the argument for diversity. And of course, this picture is a very nice picture, depicting this very clearly. We can all kind of see at first glance a very obvious kind of benefit here to the diverse mixture. We have these different and deeper root systems, some shallow, some deep. They're not competing with each other to such an extent. Some are feeding more at the shallows, some are feeding more at the depths. You have different species within those different root depths and architectures. We have different root exudates being pumped out by those specific plants. We have differences happening up above ground, the different, again, heights and architecture, flowering times, different nutrient content of some of those plants from a grazing point of view. Some plants will be mineral accumulators of certain trace minerals or nutrients as compared to others. And so this is better for the animal diet, a salad buffet up the top, for example. And yet on the right hand, we have, on the left-hand side, we have our monocultural stand. And, you know, we can see an obvious problem here straight away in terms of just using the resource of the soil, the resources of the soil, the nutrients that are a bit further down out of that rooting zone, the moisture that might be a little bit further down. We're not accessing, we're not leveraging that fertility inherent fertility that we have in the soil. These plants are going to be dependent on added fertility to top up that because they have these small root systems. And of course, you know, in a cropping situation, we remove all of the weeds. These are competitors. We don't want them. We want this perfect uniform stand because we don't want that competition. Those weeds will suppress the plants, compete with those plants for moisture and nutrients. But, you know, I can also see still on that left-hand side, I also still see a lot of competition. You have this complete uniform stand of plants. They're all the same plant species. They're generally all the same variety. And so that means that high degree of uniformity means that every single one of those plants, which are also in the exact same crop stage, developmental stage, they are all growing, as you can see, with those very similar root systems. They are all competing for moisture and nutrients in the same depth. They're all competing for the same depth. Because they're all in the same growth stages, they're all requiring certain nutrients as they move through those growth stages. We're just hitting flowering here. Just for example, boron, really important for pollination, for flowering, etc. So now we have all these plants, they're all competing for boron, all competing at the same time, all competing from the same root volume, from the same soil depth. So although we have this idea that, well, uniformity is efficient and we don't want all this competition, actually I still see a lot of competition happening there on that left-hand side. Whereas again, that example on the right here, okay, one of the plants are feeding more in the shallows, one more in the depth, etc. This kind of thing. So with that really comes multifunctionality and becomes, I think, also a lot of resilience. You don't know if it's necessarily going to be a wetter year or a drier year, maybe a bit cooler year or a hotter year. And if we have a monocultural stand that is maybe a bit better suited to the wetter years, and you end up having a dry year, of course this monocultural stand is not going to thrive. Whereas if at least you have a mix here, half of which are suited to cooler years, half of which are a bit better suited to the warmer years, depending on whatever way the weather goes, it doesn't matter, you're going to have something, some percentage of that mix is going to thrive. So you can we can observe these increases in biomass productivity through this greater diversity. So then that leads us to this question, well, what is the optimum number? How diverse do we need to be? What is the optimum number of plant species that we need in a mix? Has anybody got any thoughts? What's the magic number?
Are you talking, because I've heard Tristan talk about plant family is more important than plant species, so there are different varieties of the same similar grasses have different families.
Excellent answer. Four at a minimum? Yeah, sure. Okay. Does anyone else want to raise? Do we have anyone else want to raise you? See you and raise you? Very good answer, though. No? No one bring brave? 80? 20. Okay. Good. I like that answer too. Up to 200. Great. I like it. I like it.
I was going to say that the upper limit would be more about the practicalities rather than how many, like if you just had.
Oh, you guys are a very switched on crowd. Absolutely. You're absolutely spot on. You're right. You're both absolutely spot on. It's not just diversity per se. It's not diversity for the sake of diversity. It's about functional diversity, and plant families is a way to kind of conceptualize that. Or just plants providing a specific, different, unique function to the soil. And yeah, there has to be a top end of terms of what's practical too. I agree. The key point here is that it is not the benefit of diverse systems is not the fact that they are diverse. It's not that the more is better, the more the merrier. If 4 is good, 20 is better, then 200 has to be better. If its diversity is the mechanism of the benefit, then 201 has to be better than 200, which of course it's not. We're at that point. Now, it's about the function of the plants. If you add another plant that has a different function and provides a different function, it's now a taproot. We have all fibrous roots. We have now added a taprooting plant. We have added a different function. Oh, now we're going to add a legume. Well, we're bringing nitrogen in. Now we're going to add something else that's good at solubilizing phosphorus. We're adding functions. And when we add the function, the mechanism of the improvement for the diverse mixtures is the more functions. So to your example, if I had a choice between a mixture, a fodder mix of 8 legumes versus a 3-way mix of a cereal, a brassica and a legume, I would choose that 3-way mix over the 8-way mix of legumes, because we have different functions, different families could be also another way to view that. So yeah, that's exactly right. So I think diversity is good, and we talk about it in that general sense, yeah, the more is good, but the key to that is adding additional function. You know, as much as I also agree that diverse stands are good, we don't have to overly complicate the thinking either. I agree. If you can hit 3 or 4 or 5 kind of species, some of those simple mixes, I think, are still great. It doesn't have to be these fancy 10, 15s, 20s, etc. Although, fine, if that's working for you, fine. But I think even some benefits around 3, 4, 5 is also fine. And also, if I had a choice between none and 1, hey, I would say there's nothing necessarily wrong with 1 either, in the right context. You know, okay, there was 2 there. You can see there was some maize there, but we did have 2, but now the maize is gone. We have 1. We have monoculture on the left. And I think that looks fantastic. Much better than what's on the right. Okay, so again, just the right context. I'm not opposed to 1 either. I think it has its time and place, has its right purpose and context. No problem. I think that's still doing excellent work for this soil, capturing the last of that autumn. This is from Europe, capturing the last of the end of the season sunlight, keeping that soil covered. As the winter, autumn rains will move in, preventing some of these issues, keeping those roots down, root channels, water moving down instead of water ponding and pooling like you can see here. So again, just I wholeheartedly support all of that discussions about diversity, but you know, like time and a place, I think it's okay too. I would rather, the important thing is also keeping that soil covered, living roots. So now let's kind of dive into, I'm going to hit some science here. I'm going to throw a bunch of studies at you just to kind of bounce some ideas and interactions off you around all of this. There's a good body of work looking at some of these diversity interactions and benefits that can kind of come from that. So I'm just going to kind of do a little mini review of some of those for you all. And actually this one, I'm not going to read this whole thing. I just wanted to point out what you guys already touched on already. It's this emerging concept of selecting cover, in the context of cover crops, but species with functional complementarity rather than simply increasing the number of species, as you so excellently pointed out. This is that key message. It's not just about the species, it's about functional diversity or identifying symbiosis or plants that have complementary interactions and growth habits. So let's have a look a little bit just to set the stage about a little bit on biology. Well, of course, we're all very interested in soil biology these days. Let's have a little bit of a look of how plants can change the biology, and then we'll move into some other soil properties. And I just want to point out here this good review study. This is a meta-analysis. A meta-analysis meaning it's just a study that reviews lots of other studies. They don't do an experiment themselves. They're just reviewing other people's experiments and bringing them all together, reanalyzing them, and trying to find some broad trends and patterns in these types of things. So we have a meta-analysis here summarizing 60 other studies looking at the effects of cover crops on soil biology. And they find that in relation to, in the context of cover cropping here, that cover crops help improve soil microbial abundance, and so the microbial numbers, the numbers of organisms, by 27%, improving the soil microbial activity by 22%, and increasing the soil microbial diversity by just 2%, 2.5%. And this is the point I just want to zoom in on. We also have this slight, it's not quite right, we hear this often, that more diversity above ground encourages more diversity below ground. And this is not quite right. Soils are just very diverse ecosystems. They are highly, highly diverse because of the microorganisms. Incredible diversity, genetic diversity of life in the soil. Soils just are very, very diverse. And by adding more diverse plants on top, you're not necessarily increasing that diversity. Okay, maybe a tiny bit, but it's not really the key driver, it's not the key mechanism. What you're really doing with more diversity of plant species is simply just activating or waking up a greater diversity of those organisms. There are a huge diversity of organisms in soil, but many of them are dormant, sleeping, dormant. And by increasing the plant species diversity, we are simply activating, waking up more of those organisms, interacting with them in an enhanced, improved way. It's not really that we are increasing the diversity of those organisms per se. Okay, so let's look at just some examples. I'm just trying to build a picture here to show this point that different plant species have different specialities, ways in which they grow in the soil, and therefore can bring unique and specific benefits. So this was a study looking at soil structural genesis, so improvements of soil physics, some of the structural properties. And we're comparing four plant species here: clover, black oats, phacelia, and tillage radish, looking at how they improve the soil structure and some of the microbial properties. So I'll just read you this quote here. Black oats developed the greatest soil pore connectivity than any of the other species throughout the growth period, whereas phacelia actually decreased both the porosity and the pore connectivity. So here we're saying that, okay, black oats, nice fibrous root system, that oats were very good at improving the structure, and phacelia did not do that at all. In fact, they don't have that nice big root system. They didn't improve soil structure. Of course, phacelia is great for other things. It's beautiful flower, purple flowers. The bees love it. Great for attracting in the beneficials, etc. Has other functions, but it's not necessarily a builder of soil structure. However, the microbial community under that phacelia was notably different from the other species, with a greater proportion of fungal markers. So here we're highlighting that, okay, phacelia maybe wasn't the best at improving soil structure, but it did encourage some of the beneficial fungi in the soil. So the obvious point is that, well, why don't I put black oats and phacelia together, and I can have oats for structure and phacelia to stimulate some fungi. And again, that's that argument of diversity. They're providing a different function, and so they could be a good team together, for example. Yep, sure. Yeah, I am sharing some examples here of cover crops. We will also do some pasture examples and cropping, so I am jumping a little interchangeably here, just cherry-picking out some examples of different plants and contexts of them interacting. But here we're generally talking about cover crop, meaning plants that are growing in the off-season after the harvest. After a cropping season is generally what I'm referring to when I say a cover crop. So plants that are growing post-season that are not necessarily to be harvested, but just there for soil improvement. Now, that doesn't necessarily matter in the context of if we're looking at just some of these root and below-ground interactions. If you happen to then come along and graze that plant, if you're a grazer, for example, well, okay, some of those benefits are still happening whether or not you graze it, or if it ends up being just a green manure type cover crop. So But yeah, that's what I'm referring to here. Yeah. Okay, another one here. We're looking at, again, the effects of different plant species. And we're looking at fungal pathotroph. This means beneficial fungi that eat pathogenic fungi. So some of the beneficial fungi that suppress the pathogenic fungi. And they found that these beneficial pathogen-eating fungi, that their abundance was positively associated with oilseed radish, with lucerne and phacelia, but negatively associated with sorghum Sudan grass. So sorghum Sudan grass wasn't very good at stimulating these pathotrophic fungi, these pathogen-eating fungi. However, when they measured things like mycorrhizal fungi and some of the other beneficial nitrogen-cycling bacterial groups, that these were very strongly associated with sorghum Sudan grass. So although it wasn't good at doing one particular function, it was achieving benefits in terms of some other functions there. And this is then a really interesting kind of study that's also looking at this. Sorry, I know the numbers are very small. I'm just going to point out a few key things here for you to catch your eye. We're not going to go through all of these numbers, don't worry. But what we're doing is comparing here is some of these kind of one-way monocultures, single mixes, and we're looking at how they affect some of the different microbial groups in the soil. So bacteria, actinobacteria, fungi, mycorrhizal fungi, protozoa, these types of things. So we're just looking at how different plants influence different parts of those soil biology. And we're comparing single species, a range of different plants here, sun hemp, soybean, red clover, hairy vetch, forage radish, oats, canola, cereal rye. Then we have some kind of 4-way mixes, and then we have an 8-way mix. And what I'm going to do is just circle out for you, this is after 2 months of these plants growing in the soil for 2 months, and then they're looking at the effects of on the biology. And you can see here what I've done is just circle some of the highest reading for each of the category. So you can see that down here that these kind of diverse mixes seem to be quite beneficial at stimulating the 8-way mix here, the overall microbial activity, some of the bacteria. Okay, this 4-way mix was particularly good there, but not necessarily always the diversity was better. So here you can see sun hemp was actually the best plant to stimulate this particular group of bacteria, actinobacteria they're called now. You can see that oats was very good at stimulating the fungi, non-mycorrhizal fungi, saprophytic fungi, carbon-degrading, carbon-cycling fungi, as well as our mycorrhizal fungi. So oats were very good at stimulating fungi. So it wasn't always that the diverse mix was better, that some plants had a bit of a specialization there and were able to stimulate those different groups in a different way. Now that's at 2 months. They then repeat this at after 9 months, so more time of these plants then growing in there. And let's have a little comparison again. And you can see generally there has been a little bit of a shift down towards these more diverse mixtures with a bit more time, because sometimes benefits or interactions do take time. This is one of the big challenges with some of these short-term cover crops, off-season cover crops, is that often they don't have enough time for really expressing some of their benefits and gains. And so over the longer term, you have more time for some of these interactions to unravel. Yeah. Okay, so we can see this little shift here that, for now, for some reason, the canola here was best at stimulating these groups of bacteria, and we see that the vetch was particularly good at stimulating some other fungi. However, there's now quite a few down the bottom here. We can see that the 8-way mix seemed to have some bit more benefits with a little bit more time. So just kind of trying to bring in a little bit of that context there. So the key point then here is to say that when we start this discussion about diversity, even if we jump from 1 to 2 and start looking at 2 root systems that are interacting, the core of the idea here is that each plant species, has its own unique genetic makeup, the way in which it grows, the way those roots grow. It needs certain nutrients that it requires. It will release very specific root exudates unique to that plant species that will wake up and activate very specific groups of microbes who will cycle some nutrients, bring various benefits, and help that plant grow. And so the plant has the tools that it has. Those tools are determined by its genetic makeup, which then those genes can be turned on, expressed to trigger specific benefits or changes or root exudates or functions in the soil. And the point is that, well, if we simply just add another plant species, a second plant species, an example of a legume here, well, that legume is, of course, a very different plant, has a different form, different root architecture, produces different root exudates than what the cereal would produce. And when we bring them together, we have this process of root interaction. And that really simply means that the legume is now releasing specific exudates into the soil, activating specific groups of microbes that the cereal cannot activate, that the cereal cannot work with. It is now growing in that environment with that legume, and the legume is doing the work and activating microbes that now the cereal can benefit from. But the cereal on its own could not benefit from that. It could not activate those organisms itself, but once we bring them together, it can receive the benefits of the organisms that the partner is activating, and vice versa. So of course now the legume, if it was the legume in a monoculture, now the legume has a cereal friend here. It's working on its own interactions, and the legume can benefit from that. And that's the core of the idea. We could open up this discussion and say, well, okay, then there's a potential there for synergy, 1 + 1 = 3. What we can achieve is greater than the individual components of each of them themselves. And that's kind of what we're looking for in terms of some of these synergistic interactions. So the question was, how does fertilizers then impact some of these interactions? And yeah, that's a big question. That's a good question. That's a big question. It can have both impacts. It can be positive or negative. It's not to say, I would say, that fertilizers are always negative. They're not. We often hear this, this discussion. I wouldn't totally agree with it. I think it's about the right dose, the right rate. If you over-apply fertilizers, yes, they can be more damaging. Small amounts, I would say, not always that they are bad. An example, it's not just plants that require those essential nutrients, nitrogen, phos, all the micronutrients. The microbes require those nutrients too. And so if a microbe was growing in a nitrogen deficient situation and you added a bit of nitrogen fertilizer, you would actually increase their function, increase their growth, because you've overcome their nutrient deficiency. In the same way that we use fertilizers to overcome plants' nutrient deficiencies, you can do the same with the microbe. They require nutrients too. So in some instances, fertilizers can increase microbial activity and growth if those nutrients are helping overcome a limitation for them. Now, however, that said, yes, over-applications can definitely be disruptive in the same way that you know that if we apply nitrogen to a legume, those nodules get lazy, it doesn't fix nitrogen. In that same way, we can extend that thinking to this too. If you're applying nutrients that makes it easy for the plant to obtain, then it won't maybe invest so heavily in root production or some of these root exudates. It can short-circuit that interaction. So yeah, there can be some negatives there. But it's not inherently bad. It's true that all fertilizers are bad, and that whole kind of discussion. I don't quite agree with that. So that's what we're trying to exploit, harness. And okay, let's not pretend just because we increase diversity, just because we've gone from 1 to 2, that we can say for sure that's going to be also a positive effect. Plants can be competitive and they can be collaborative. It can go either way. Just because you're now adding 2 doesn't surefire mean that it's going to be better. Okay, some plants release root exudates, some plants interact in negative ways. They're not good partners. They don't necessarily always enhance each other's growth. So that's also, I will acknowledge that. And we have knowledge gaps in this for sure. If you look at the whole field of intercropping at the moment, companion cropping and intercropping, it's definitely taking off in many parts of the world, especially Canada where I'm based, and quite a few countries in Europe. Still a little bit behind here, I would say, in Australia, but there are some classic examples of plants combinations that do seem to really work. The classic things like the peola, the pea canola, or pea and oats, peas and barley, wheat and beans. There are some examples that we know seem to work very well. There are others who could go on, but so we have, I think, enough baseline knowledge. But for sure, we also know that even varieties have an impact here. So some varieties are better suited with some of these positive interactions than others. And some of the farmers that I know in Canada who are intercropping, for example, are observing this. They're finding that some of the older varieties actually seem to lend themselves a bit better for intercropping systems. They have more of these positive interactions. Some of the more modern varieties, of course, which have been bred under a monocultural system, they have bred to be highly productive. Maybe they're not have been bred for some of these synergistic interactions. They may not be the best ones for this process. So I think that there's a lot of opportunity here, most certainly, but I think there's also, we've got to do a little bit fine-tuning and breeding and some research on how best to harness some of these interactions between the plants, because they're not always positive. Okay, so here's another example, just looking at diversity on, again, just to close up some of the microbial discussion. Again, we have a meta-analysis here. We're reviewing kind of 106 different studies, and they show that microbial biomass, bacterial biomass, fungal to bacterial ratio, microbial respiration all increase, all these biological properties all increase with increasing plant species diversity. And the effects of that are consistent in forests, in planted grasslands, in croplands, and even in planted containers. So again, just highlighting that the more diversity there affects, the more diverse systems seem to generate better biological functioning and activity in those contexts. Okay, what about physics? Here we have an interesting example, again an intercropping one, just moving from 1 to 2 and looking at how that impacts aggregation, pulling together the soil particles, improving this physical structural benefit. And they show here that soil macroaggregates in the intercropping systems, this is in maize and faba bean, again, just moving from 1 to 2, just having 2 root systems instead of 1, that we increased aggregate production anywhere from 15 up to almost 60% increase in aggregation. And that was primarily driven through microbial interactions, that the diverse root systems, the two-way root systems, stimulated microbial groups more effectively, and they are the ones kind of gluing and sticking together the aggregates, improving that soil structure. And when we stick aggregates together, we create pore space in between. We have porosity in between, which is very, very important. So again, small change in the system design, but potentially big impacts in aggregation. And this is what I'm talking about when we refer to aggregation, just pulling soil particles together, stabilizing that soil, gluing that soil together, stabilizing that soil, creating space in between. That space is key for when we do get rains. That moisture, we want it to go down, not horizontal across the top of the soil. We want it to infiltrate, so getting that moisture down into the soil, but also good porosity for gaseous exchange, for air, oxygen to come in, for CO2 to come out, for that whole gaseous cycling as well. Okay, another example in, again, grasslands. This is both in a glasshouse and field study. We show that high plant diversity in grassland systems increases the soil aggregate stability, a vital structural property of soil, and that roots were a key driver of this. So again, looking at various structural properties of the soil, finding that the diverse systems with their diverse root systems, the direct activity of the root system there, driving some of these improvements to the soil physics, to the soil structure. Okay, so a couple of examples there from physics. Let's move on to a little bit on chemistry and nutrient cycling. And here we have a really nice visual, fascinating example here. We're looking at, again, a faba bean root system and a maize root system. And you can see that these two plants, they're releasing their various root exudates. And you can see that the beans, the legume here, is driving the root system, the rhizosphere, to be more acidic, dropping the pH, making it more acidic, whereas the legume, the cereal, the maize over here, having more of a neutral, changing the pH to be a bit more of a neutral range. And again, this is just pointing out those differences between plants. Some are different, therefore they have these different specialities, and the opportunity to bring them together so that they can benefit from that. Legumes, they require molybdenum for that nitrogen fixation process. Molybdenum is a very important nutrient for the rhizobium bacteria to fix nitrogen. And molybdenum is more available under more neutral pH conditions. So that bean grows into the rhizosphere of the maize, where we have more neutral pH, where molybdenum may be a bit more available, well, the legume can access that. Or similarly, if the maize is trying to get some trace elements out of the soil, well, this acidification of the soil is going to make some of the trace elements a bit more available, and that maize could potentially benefit from that. So we have this interaction there where we can exploit these specialities of the various different plants. And this is all we're just looking at in these charts here. You can see we're just looking at the production of some of the organic acids that help to strip minerals, make minerals available, access them off the soil and make them available to the plant. You can see that the bean is producing a lot more of these organic acids than the maize, and consequently the bean is also accumulating much more phosphorus in its tissues than maize. And legumes, they have a very high phosphorus requirement. The process of nitrogen fixation is very energy intensive. It requires a lot of energy to do that for the bacteria. So the plant has to help provide some of that energy. And this is why legumes have evolved mechanisms to acquire phosphorus from the soil, because they need a lot of phosphorus to produce that ATP molecule, that's the energy currency of the plant, to provide that energy. So legumes have got all sorts of specialized ways and means in which they access phosphorus from the soil. And so their tissue generally is a higher phosphorus concentration than many other plant species. And these are some of those mechanisms. It can be, as I just mentioned before there, releasing organic acids, releasing hydrogen, just acidifying the soil, but also other groups of special root exudates, a group here called phosphatases. These are special enzymes that are root exudates that are excreted from plants, which can help to unlock organic phosphorus, some of the phosphorus in the organic fractions, and liberate that phosphorus for the plant. Now, plants can do this directly, as you can see up the top half, but they can also do it indirectly in concert with the microorganisms of the soil. So they can feed the fungi, support mycorrhizal fungi. Again, legumes are good mycorrhizal associators, supporting mycorrhizal fungi, let the fungi strip that phosphorus, access that phosphorus out of the soil. Feeding the bacteria, letting those bacteria, some of those organisms are good phosphorus solubilizers as well. So the plant can do it directly itself, yes, but it can also do it in partnership with some of those organisms. And then this point, a really good example here on your symbiosis kind of discussion here. This one is a really interesting study, just published last year. And here what we're doing is just measuring the root exudates, again, from maize. We're looking at the organic acid production, some of these different root exudates. And we're looking at measuring the root exudates from the maize under monoculture, and then a couple of different contexts with a friend, with a partner. So we combine maize either with soy, with lupin, or with mustard. Now, lupin had the biggest effect here, but I'm just pointing out what we're doing here is we're just measuring the root exudates from the maize, whether that maize exists in a monoculture or whether that maize has a lupin companion nearby. And what we are showing here is that it's not just, I gave these examples of legumes being good at solubilizing nutrients, and I gave this example here of that the cereal will benefit from the action of the legume, or the legume can benefit from the action of the cereal, those types of examples, where they're kind of working together, so to speak. What we're actually showing here is that compared to maize as a monoculture in the brown, that if we just simply have a lupin companion nearby, that then the maize itself actually changes its root exudates and produces more of these organic acids to solubilize phosphorus, that we are actually changing the expression of the maize itself. I'm not talking about the lupin doing the work here. I'm saying that just having the lupin as a companion, the maize can detect that the lupin is there, and correspondingly it will change its root exudates and produce more root exudates and cycle nutrients in a different way than which a maize monoculture will. And so, fascinating example highlighting that, okay, it's not just these direct interactions, that there can also be changes in function. Plants can turn on different groups of genes in their body that they cannot do under a monoculture system, but that can seem to be activated when you have some kind of a partner there, some kind of an interaction there, which I think is fascinating, shows us there's a lot we still don't know, and there's a lot of opportunity there. consequently to potentially harness some of these diversity effects, some of these functional diversity effects, some of these types of interactions. So yeah, there's a very good paper that's looking, I touched on this a little bit in my example earlier of not having, I was talking about in the context of intercropping and having the varieties that are best suited to that. So we have some examples like that saying that that some varieties are going to be better suited, so that therefore the genetic makeup of those can be steered towards it improving those interactions. But if we step back to that in terms of what you're talking about, if that, like what I was also demonstrating here, if that plant was growing in a monoculture previously, where it is expressing, fulfilling its life cycle, expressing that reproduction, producing that seed, you know, all of the genes that get turned on during that growth process, during that growing season, ultimately are the ones that kind of get upregulated, activated. So the plant will have a memory of that essentially in the seed, versus if that plant was growing in a more diverse mixture where some of these types of interactions are happening like this, and therefore we're not changing the answer to your question is really this whole thing of epigenetics. We're not necessarily changing the genetic makeup of the progeny, but it is through epigenetic changes. So the genes are still the same, but which genes become more active, that environmental interaction of that can indeed then be different. And it's, I would say, it's a very, your question is a great one. It's a very untapped area. We haven't really gone deep into identifying how plants in that next generation are different from if they are grown in a diverse mix versus a monoculture. I think we have the examples there where it's the mechanisms are in place, but I haven't seen any concrete kind of work showing or exploring what those differences may be. My suspicion is that there would be a difference. I think we have the mechanisms to explain that and why, but no, I haven't seen a kind of a firm paper on this. In terms of then this discussion about, you know, improving nutrient use efficiencies and using the discussions around reducing fertilizer use and leveraging the fertility that we have in the soils, this is our kind of the argument here. This is the opportunity that we have to try and be more efficient. We know that many of our fertilizers are rather inefficient. Nitrogen, of course, very reactive. It can leach through the soil, volatilize off into the air. It can be lost from the system. So generally, nitrogen fertilizers hover around about a 50% nitrogen use efficiency. About 50% what we apply goes into the plant. The other 50% is, some of that is lost. Hence why we have this whole discussion about nitrogen in the atmosphere and in the environment at the moment. Not all of that nitrogen is necessarily lost just because it's not taken up. Some of it is consumed by the organisms in the soil, embedded into the organic matter in the soil. So some still remains in the soil, but some can potentially be lost. Phosphorus also very inefficient. Because it has a tendency to lock up. Our phosphorus fertilizers have a tendency to lock up in the soil. It's not so reactive. Phosphorus is not lost quite so easily, but it can be lost through things like erosion. Physical removal of that phosphorus, for example, then can be a problem. So the point being is that generally our fertilizers are rather inefficient, and we do have an opportunity to do better, to use less of those, to try and improve those efficiencies. And part of that discussion, and I know that it opens up into this whole debate about soil mining and all this kind of discussion, but which we can also touch on, but part of this is just to say that in soils, and this will be a little less relevant for those of you that do have very light sandy soils, your potential for this is a bit smaller than others that have a heavier soil, but in all soils, you have three kind of main different pools of nutrients. The smallest fraction is what's actually soluble and generally considered to be plant available. You have a very small fraction of what's in the soil water, in the soil moisture. We then have this other fraction called the exchangeable. This is your medium-term supply. This is all the nutrients that are kind of attached to the soil colloids, to the soil surfaces, to the clays, to the sands, to the silts. And of course, a sandy soil has much smaller ability to hold on to nutrients than a big, heavy clay soil, of course. So again, much greater nutrient reserves in clay soils versus sandy soils. So these are your exchangeable. Now, both these two, the soluble pool and the exchangeable pool, they're both fairly plant available. If it's in the water, yes, the plant can take it up. The plant can exchange these. It can release some of that hydrogen or the organic acids I talked about earlier and exchange the potassium in this example, exchange the nutrients. So both of these two fractions are fairly plant available. However, and generally when we have a soil test, usually these are the fractions that we're measuring most of the time. But you do have a third, much larger, the largest pool of nutrients in your soils, which is kind of what's called the total pool. And this then refers to all those nutrients that are either locked up, they're present, but they're just not available. They're kind of locked up with other nutrients. So under acid conditions, that phosphorus, for example, might be locked up with aluminium or iron phosphates or aluminium phosphates, these types of things. Very insoluble and unavailable, tightly stuck together nutrients that have poor availability for the plants. Some of those other total pools might still be part of the clay layers or trapped within the soil particles, or again, part of that parent material that still hasn't fully kind of weathered or become available. So point being, you have a big pool there, and it's not to say that it is completely inaccessible. You can unlock these total pools of nutrients, but the key to that is some of these biological interactions. And yes, plants and plant roots and plant root exudates can do it themselves, but all of the biology, particularly the microbiology, they can do it so much better. Fungi, bacteria, they release these very specialized compounds into the soil, these microbial metabolites, enzymes and things that are very specialized at unlocking, breaking apart that bond of phosphorus and iron, for example, chelating compounds that grab onto that phosphorus and remove it off the soil particles, these types of things. So the point here is that, yes, plants can do a bit of this function themselves, but microbes can do it so much better, just for the simple reason that microbes exist in soils in such greater diversity. Because you have such incredible genetic diversity of microbes in soils, with that genetic diversity comes a diversity of functions, a diversity of specializations, a diversity of modes of action, etc., etc. Okay, so microbes can do it much better. They have more tools in the toolbox to access and cycle. So that's really the argument here. It says, okay, in order for us to be more efficient, to reduce our dependency on fertilizer inputs, we can try and tap into some of this reserve, and improving biological function in the soils is key to that, and that's where plant species diversity can also sit, because the plant species diversity can enhance the functionality of those microorganisms. Again, doesn't really increase the microbial diversity, but it does increase their activity, their functionality, these types of things. Okay, and that's kind of the argument. Now, we could then go on, and we don't have to kind of labor on this, but have that debate about, well, are you mining the soils? And if we don't put replacement fertilizer down or crop offtake, all of that discussion, are we mining the soils? And, I'm not a guy that's a no-input guy. I think that we definitely are very inefficient with many of our fertilizers. We clearly can be using a lot less than we currently do. I think that is for sure. Whether or not we can be using zero, I don't know. Definitely not, I think, in a sandy soil, in a heavy clay soil, in a grazing on a heavy clay soil, yeah, potentially. I think you can have very good opportunity to use minimal inputs in that kind of a context. In a cropping situation on a sandy soil, obviously it's going to be much harder. Cropping, you're going to see a lot more offtake and removal. Light sandy soils have much lower potential to hold on to nutrients, and they have less of these total nutrients in them. So I think there's some context there, but do I think that we could definitely be moving in that direction? Absolutely, yes. We could be using a lot less fertilizer than we currently do. Key to that is improving this, cycling of the totals, as you can see here, and using plants to achieve that. But also part of that is choosing the right plants that can even just have deeper roots to access some of this. Think about those total pools of nutrients that we were just looking at, and then think about it in this soil volume. You have a significant pool of nutrients here that these plants are just not accessing. Not only just that physical volume of soil, but then the total nutrients within that physical volume of soil. And this is the argument for reduced fertilizers. Okay, if we use diversity, particularly with deeper roots, we can start to access the full profile and access a much greater amount of the soil's inherent fertility than these types of systems, which we have these piddly plants with small root systems that become very dependent on fertilizer inputs because the roots are not leveraging all of that fertility, potential fertility that's there. So I think that's kind of the opportunity, diverse roots and accessing the totals. But I acknowledge, I'm not saying we maybe can use none, but we can certainly do a lot better. And I think that in certain contexts, we could definitely be using none, heavier soils, grazing type systems, potentially. So the question was, can we change sandy soils? Can we improve sandy soils? And that was the essence of the question. And can soil become more clay-like? Yeah, okay, so yes, you can improve sandy soils, but key to that, I mean, you can't make them more, you could physically add clay, I mean, that is an option, and there are some materials like, for example, zeolite, if you've come across this one, which are basically like that, these volcanic clays that are permanent additions to the soil that can then, they offer a much bigger sponge that can hold onto moisture and nutrients than what the sandy soils can do. So you could add inputs like that. But key to really improving sandy soils, we have to, on some level, we have to accept that they have a limitation or a top end in which we can change them. But yes, we can improve the organic matter fraction. And that's, I'll also acknowledge, is not easy, it's difficult. Well, I think we'll talk about this later this afternoon, but that's the key thing you need to more so focus on with the sandy soils, trying to improve the organic matter of that soil. Yeah, building organic matter doesn't happen overnight, for sure. It is a long-term game. And, that said, I think within a few years, you can see if you're moving in the right direction. You can see indicators of progress early on. But in terms of making kind of bigger, meaningful change, yeah, I agree. It is a longer-term thing. This is being hotly debated now, and I think maybe we'll carry on this conversation when we're outside, because I want to talk a little bit about what I alluded to earlier, roots and root exudates, and some of this kind of discussion about how these two things kind of build soil organic matter and help. So we'll have that discussion maybe outside, but it is a long-term game. You're not going to do it overnight, but yes, you can improve sandy soils, and the key is the organic matter. And when you improve that organic matter, you can then increase that sandy soil's nutrient and moisture holding capacity. So you can add clay or clay-type materials. Generally, that's can be quite expensive. Hauling that in and spreading that, etc. It's possible, and it is done in high-value veg-type situations, but generally not in other contexts. So the key is really just trying to use plants, better grazing, use your animals to try and build that organic matter in those sandy soils. So the question was that is the statement true? 95% of the world's biodiversity exists in the soil, and I don't know what the exact number figure is, but yes, this is what I was alluding to earlier, that soils are incredibly biodiverse environments. Soils and coral reefs really are the top two. And yes, this is the point, that it has incredible genetic diversity in soils, but it's all mainly through all of those microorganisms, primarily the bacteria and fungi, for example. And yes, they exist there in just incredible diversity. There are tens of thousands of different species there. And this is therefore why in a tiny little gram of soil you can have a huge number of different species compared to then larger, more macro level terrestrial ecosystems where we have, sure, forests are very biodiverse, tree systems. Yeah, in your forestry situation, yeah. So yes, we talk about the incredible richness of diversity of forest ecosystems in the tropics, and some of those different plant species that exist. Incredible diversity for sure, incredible diversity of the insects that then live in those systems. But all of this pales in comparison to the vastness of the diversity of microorganisms that exist in soils. Whatever the exact figure is, I don't know, but the principle there is absolutely valid, yeah. And thank you, your prompt. I did want to mention biochar as well as another example like zeolite, but biochar is also a permanent addition to the soil, so it's not carbon that's going to kind of cycle and break down and be used. It's a permanent addition, and so it would also have relevance for sandy soils to try and improve them and increase their nutrient moisture holding capacity. Yeah, that's right. The delving thing is interesting. I see, I don't know lots on this, but I see it's becoming a bit more popular, or there's some positive studies showing that it's beneficial. Obviously, it's a very aggressive, intensive process, but it's a short-term kind of intensive treatment to try and then bring more long-term sustaining benefits. So yeah, I follow that with interest too. I think, I can't say I have a deep knowledge of this, but from what I've gleaned, it seems to be beneficial, yeah. And for that reason, you're just trying to bring a bit more clayey type materials into the soil, yeah. Okay, a couple of other quick examples just to maybe round off. I was just going to, I won't dwell too much on this, I want to move on. Let's do a couple of little pasture examples to close off. But I just want to highlight that even in this discussion of nitrogen also, of course, nitrogen, fertilizer prices at the moment, hot topic, and we have a lot of, therefore, motivation to try and be using less, be more efficient with our nitrogen inputs. And if we want to do that, you know, there's an obvious opportunity is to use legumes, to lean on our legume friends to help supply some of that free nitrogen that is in the atmosphere, thousands and thousands of tons of it hovering over every hectare. We have lots of nitrogen there. It's just plant unavailable, and our bacterial friends that work with our legumes can, of course, unlock that and deliver that into a plant available form. And I just want to quickly point out that we can also share, legumes can share that nitrogen in real time in the current growing season. It's not just when legumes die and decay and provide nitrogen for the next year or the next crop in rotation or the next growing season. There can be real-time sharing, and that happens through root exudates. We talk about root exudates. I've touched on them a bit already. We often talk about them, the sugars and the carbohydrates that plants release as root exudates. Yes, they do release those, but they also release amino acids as root exudates. They're also among the most common root exudates. Sugars, carbohydrates, organic acids, and amino acids. And amino acids are organic forms of nitrogen. And so the plants release lots of amino acids as root exudates as well. And indeed, those organic forms of nitrogen that the legumes release can indeed be scavenged by a non-legume, particularly if that non-legume is a nitrogen-hungry plant, like a cereal, like a grass, like an oilseed. They can indeed steal those amino acids as they are being excreted by the legume. So you have basically direct sharing of root exudates. And then if the two plants are both mycorrhizal associating through this common mycorrhizal network, we can also see sharing of, again, predominantly amino acids through the mycorrhizal fungal network and delivered to the non-legume. So we do have these real-time sharing opportunities of root exudate scavenging or sharing through the mycorrhizal fungi in that current season. And again, I would just say that we have the evidence that this occurs, and we have some interesting work in the cropping context looking at what a legume can supply, what percentage of nitrogen can it supply, and it's highly variable. It really depends. It can make at least a contribution, I would say, so it's a worthy thing to explore. But I would, again, that point about breeding and varieties, we know this works, this mechanism exists, but it has not ever been bred for. It has not ever been fully harnessed. Some examples that we do know already, if the two varieties, both the legume and the non-legume, if they both have more lateral roots, more kind of horizontal roots, then we see a better, stronger effect of this nitrogen sharing. So, for example, lucerne, it's a powerful nitrogen-fixing plant, but it has those very deep roots. It's not necessarily great at sharing it in the real time. It's great for the next season, its residues, etc., but it's not a good sharer. Another example, clover. We've had some studies that show that, for example, little-leafed varieties versus big-leaf varieties, that little-leaf varieties of clover are better at sharing nitrogen because they have a smaller leaf, therefore they have a lower nitrogen demand for themselves, so they can share more, versus a big leaf variety that needs to keep back more of that nitrogen to grow that bigger leaf, therefore it can share less. So these are some of the things that we know of. So my point is that the potential here, if we bred for this goal, if you want legumes to share nitrogen to help our cropping systems or to help our grasses in a pasture, we can breed them to be better sharers. And typically, again, we're using varieties that have come from, kind of to your question, overlapping a bit, we're using varieties that have been bred for monocultural legume production to be productive, and they may not be the best genetics for this sharing functionality, for example. So definitely good opportunity there, but has not been fully harnessed yet, for sure. Okay, so let's look at a few quick grazing examples to finish. And here's, again, sticking on this nitrogen story here, an example. Okay, this study is from Ireland, so take it with a grain of salt, fine, but I can still demonstrate the principle of legumes sharing that nitrogen also in a grazing setting. So here we're looking at comparing a nitrogen kind of fertilizer trial, a nitrogen gradient, and we're looking at comparing that across a monoculture of perennial ryegrass with a little bit more diverse mixtures. Mix 1 is just with that ryegrass, we added timothy, a nice deep rooting grass, and red and white clover. And in Mix 2, we've also added a few herbs, chicory and plantain. And so let's have a look. We're looking at a monoculture versus just a little bit more diversity, a few legumes thrown into that mix. So let's look at our baseline production here, 0 kgs of nitrogen under a monocultural perennial ryegrass. Okay, boom, there's our baseline production level, around about 6 tonnes per hectare, for example. If we follow this line across, just by having a couple of legumes in the mix, we have completely out-yielded that at 0 kgs of nitrogen in our Mix 1 and also in Mix 2. So let's move up again, okay, to 120 kgs. Now we've added a little bit of nitrogen to our monocultural ryegrass. Okay, now we've got around about just under 10,000 here production. If we come across to our diverse mixtures, okay, even still at 0 kgs, we have still out-yielded 120 kgs of nitrogen on a monoculture, and once again, we have also out-yielded. So let's go up to 240 kgs of nitrogen on a monocultural ryegrass. Okay, here's our production as we come across here. Okay, now we can see we have a small deficit there. If we come across to our Mix 2, however, we can see we are on par. So, okay, this is only, I would say, a small difference here based on the fact that it's 240 kgs of nitrogen here. So, okay, but even this diverse mix here, you can see it's on par. So I'm just sharing an example with you that shows that even at 0 kgs of nitrogen, with just a little bit more diversity in the mix, we can match 240 kgs of nitrogen with a nitrogen-hungry monocultural grass plant. Okay, so the opportunity there for using legumes in the system to just provide some of that free resource. If I were to answer that question a little bit generally, yeah, you're right. It is common that sometimes it might be completely sprayed off and they would establish brand new plant stands to kind of start that experiment. And, you know, you could also argue in that instance that in doing that, generally legumes that are in a pasture mix, they're not necessarily always so good at fixing nitrogen sharing in that establishment year. They do keep more of that nitrogen to establish, and then they can kind of start to share more in latter years, in second onwards years. So there are some of those subtle differences in there, but so it's a good question, but I don't know specifically on this study. It would probably be a bit of a mixture of those types of contexts, sometimes sprayed out, but potentially sometimes not. Let's have a look at some more. I was going to skip over that one, actually. That was just a nice little example of a two-way mix there. Okay, here's another one, looking at diverse mixtures. We're comparing a mono of mustard here with a 4-way mix and a 12-way mix. And they show here that carbon dioxide uptake is 2 times higher in the 4-way mix and 3 times higher in the 12-way mix. Microbial biomass, 8% higher in the 4-way mix, 18% higher in the 12-way mix. Fungi and actinobacteria were particularly the most responsive. So here we have a nice example saying, okay, from that step from 1 to 4 to 12, yes, we are increasing the carbon dioxide uptake, so pulling more carbon out, growing more biomass, feeding that to the microorganisms, growing more microbial biomass. So the question was, you know, with fertilizers, we see this plateau, the point of diminishing returns. For more input, we're not getting any more return. And within the biological context or plant species diversity, does similar types of things, does a similar plateau exist? It's a good, I think it's very hard for me to answer that question. It's a lot of different contexts and variables that may be at play, context by which maybe you asked that. Again, I would come back to that study I shared about the maize with the lupin, highlighting that it behaved very differently just by having that extra plant species there. And that brings up this difficulty in answering that question, is that as we increase plant species diversity and we change some of these similarly biological functioning, microbial functioning down below, it is clear that it is inducing changes that are over and above what we really know about or can predict. It's inducing very different and specific changes here. And for some of you that I'm sure have heard Christine Jones talk a little bit about some of this type of concept too, in relation to that. So it's hard to say. I would say that over time, plant species diversity, of course, isn't always maintained either. It can change over time, often declining. So you may not, I wouldn't say that we could guarantee that you will have this self-perpetuating, forever positive, virtuous cycle. You can potentially have that, I think, with good grazing management. If the system is functioning, that's what we're trying to achieve. But, you know, let's be honest, sometimes some plants struggle with persistence. Maybe they get grazed out more heavily, and over time that mix can actually lose some of the diversity. We definitely know that some other studies which say that the effects of nitrogen fertilizer can enhance that. So nitrogen typically then selects for some of the more grasses to dominate, and we do lose a bit of plant species diversity under more nutrient-enriched systems. So there's a lot of kind of dynamic variables that can change in either positive or negative, and therefore maybe a little hard for me to answer that question. I think it could go, I think potentially yes is my question, but I wouldn't say that it's also a guarantee or a surefire. I think it's quite a mixed bag there. Yeah, that's a good question, a tough one. Okay, so I just want to read you a little summary here of this one. I thought it was just very interesting. Not too many studies that I've come across have kind of measured the CO2 uptake thing. I think this is quite interesting, showing that the more diverse mix is also drawing in more carbon dioxide. So the results of this study suggest positive impacts of plant diversity on carbon cycling by higher atmospheric carbon uptake, higher transport rates towards the rhizosphere, higher microbial incorporation, and then prolonged residence time of that carbon in the soil. So I think that's another great example. And what about drought? Something that I'm sure you'd also be very interested in. This one's really interesting, looking at how diversity can improve the drought resilience. And it's really just a fairly obvious point, which we kind of alluded to already. This is the point of the diverse mixtures. You're getting different functionality, different plants that have different specializations, different benefits, different modes of action. And this is also true within a drought resistance point of view, that different plants have different ways in which they can tolerate and deal with dry conditions. And they have different specializations to handle that. And so by increasing the plant species diversity really just means that you're going to hedge your bets and spread your eggs across, not all in one basket, with some of those strategies in order to optimize production under dry conditions. And the kind of a nice example summarized here in this little image, you can see that this plant species, its specialization is dehydration avoidance, meaning that once it's established and gets moisture into the plant, its mechanism is very specialized at just holding on to water in itself. It's not losing water through evapotranspiration, etc. That its specialized mechanism is to get water into itself and then hold on to that. Now, therefore, it can maintain productivity in the early stages of drought stress because it's holding on to that water, maintaining productivity in those early stages. But that benefit over time dwindles. It can only hold on to that moisture, so to speak, for a certain amount of time, or as the soil ultimately gets drier and drier, it's not so effective in these more extreme dry scenarios. Whereas you would have different plants who more so, rather than being able to avoid that dehydration effect, their specialization is just that they have a very high tolerance of growing under dry conditions. So at this point in time, this plant species and its specialization can kind of kick in more in the latter, more severe stages of drought. So by having if you just had one of these, of course, you wouldn't be optimizing the opportunity of harnessing each of their specializations by having at least, for example, the two here. We could extend this thinking, but having the two, you've got one that's going to function better in the early stages of dry and one that's going to function in the latter stages of dry, overall evening out the production. You know, I think there's a lot of learning we can still carry over from what's happening in the Northern Hemispheres, and I think I'm sharing, some of the studies I've been sharing have come from there, and I think there's still fascinating learnings in terms of some of those interactions that I've been trying to share. But in terms of cover crops and summers, summer covers, of course, I agree, it's a huge challenge here with the moisture limitation, is that, you know, the system that we currently have is geared around summer fallow, preserving moisture, so to move into that cropping season. And so, I acknowledge that. I think that, therefore, we can't translate exactly everything that we see from abroad. I think there are some principles still at play that would still have relevance, but I think that you have to modify the practice according to your moisture constraints. Now, that might mean that might mean that cover crops are not possible in this context, but it might just also mean a rethink of those. Maybe the plant population can't be as, the seeding rate can't be as high. We don't want to be drawing down too much of our moisture reserves. So maybe the seeding rate or the choice of species needs to be modified. I would like to think that there is some potential for this. I think that we haven't really cracked it yet fully, but I would like to think that there's some still potential for this. But yeah, you're right, we can't just follow the same rules. I think we have to learn which of the species, what are the types of seeding rates that we could maybe establish some summer cover without depleting our soil moisture. And it's just then this tricky part of that is that if you then also do that in a different system, you know, a system that is using more diversity, is there some other benefits or changes that occur in these systems as compared to our, you know, some of the kind of, to your question about, well, what was the history of that Irish study there? What was the field doing before? Of course, as you appreciate, you know, some of the trials and studies that we do, they operate in isolation at a research farm that's not holistically being managed in a particular way. That then if you took that study and planted it into a real-world working farm with the rotations and the diversity and the years of these effects, would the study results be the same as when we do it on a research station with a completely different history of that field? You know, and so is there the potential that some of those studies that say cover crops don't work here, being done in that kind of a context, that maybe there could be an alternative view or something we could harness or learn if it was done in a different setting? So I'm hopeful, but I acknowledge we can't apply the learnings directly. Of course, we have to modify the context here for sure. Yeah. Okay, well, here's another example on this, because we could also look at it in ways to try and improve the moisture limitation, the water use efficiency. And again, just a pot trial here, but a fascinating example nonetheless. We have a pot trial of a deep-rooted pigeon pea with a shallow-rooted finger millet growing in the same pot, watering these to get them established. And you can see the finger millet, however, is growing in a little fine mesh bag. So there's no root-to-root interactions between these two. And so they get them established, and then we stop watering from the top. We impose a drought from the top, but the deep-rooted pigeon pea is able to access this water reservoir, you know, trying to mimic subsoil moisture, for example. And consequently, biomass production suffers because we impose a drought from the top. Now, they then added mycorrhizal fungi. And again, this point about the common mycorrhizal network, mycorrhizal fungi are much finer. They can grow through the mesh bag, and whereby now the two root systems are connected through that mycorrhizal fungi, which can grow through the mesh bag and connect the pigeon pea and the finger millet. And again, they imposed the drought on top, stop watering, and they demonstrated here that the pigeon pea would actually access that subsoil moisture, and it was shared through the mycorrhizal fungi to the finger millet, and we increased production of the finger millet. So there's one small example. Maybe we could also be improving our water use efficiencies. And I think this is an interesting example, particularly in the context, we'll talk about this a bit later when we go out into the field, that And that image I showed of the diverse mixture on the right and the monoculture on the left, many of our modern plant varieties, they're not big rooters. The reason that they are modern high-yielding varieties is that we have bred them to be high-yielding, meaning we have bred them to produce lots of above-ground biomass. And that comes at an expense or a cost of root biomass. So I think that there could be maybe opportunities to improve the water use efficiency with some of our modern varieties who are not deep rooters and not making best use of subsoil moisture, and potentially having a more diverse stand, actually improving that, for example. Again, we have to translate some of these thinkings across, don't we, like always. Let me just put maybe one more up in relation to soil carbon. I think, because this will be a topic that we'll come back to, I'm sure, and talk further, but just so you know, there's also a couple of good studies that show that more diverse systems can also accumulate more soil carbon, this just being one of those. So we're looking at 1,001 observations comparing mixtures to monocultures from 121 studies, and here they show that both the carbon content, the carbon stocks, are on average 5 and 8% higher in the species mixtures than in the monocultures. And again, these species mixture effects are consistent across forests, across grasslands, and also cropland systems. So again, cutting through all of that, it's a global meta-analysis here, so cutting through all of the variability in terms of different soil types, different environmental conditions, etc., across all of that variability, they're still seeing this trend that the more diverse mixtures can also accumulate more carbon. So in that context, how can we increase plant species diversity if this is, the thrust of the discussion? Sure, within a cropping situation, we can still do this just with widening the rotation, maybe bringing in novel cash crops, for example. Can we get more diverse systems in through the rotation, through cropping? Generally, the use of annuals and perennials, if you're a perennial-based system, can you integrate a bit of annuals? If you're an annual-based system, can you integrate a bit of perennials? They are different, again, have different specializations, so can we kind of make use of that, those opposing kind of plants? Similarly, same idea with summer-winter. If we're a winter-dominant system, can we bring in some summer species? They are going to be different, do different things to the soil than what our winter kind of hardy plants would do. Again, in the cropping situation, of course, the opportunity for cover crops, and to your question there about here in Australia, I think this is a similar problem in Canada compared to the US. Believe it or not, the same debate happens even across the border from Canada to US, where many Canadian farmers are also saying, well, the cover crops are not possible here in our context because we're further north, our growing season is so short. After harvest, it snaps cold very quickly. We just don't have time to even establish a cover crop. So again, similar challenges there. So what's happening in the prairies of Canada is it's a lot more companion cropping, intercropping. This is, I think, part of the reason that intercropping has become much more dominant in Canada than cover cropping, which is more dominant in the US, because the challenges that cover cropping presents in that environment. Their way to try and bring diversity in is then through the cropping phase, so with intercropping and companion crops. And then, of course, in a pasture situation, it's much more easier to integrate more diversity, maybe even trees, bringing in trees, grasses and animals. I think there's got to be an opportunity there. And also just making use of non-productive areas, the field margins, having wildlife corridors, biodiversity strips, flower strips, tall grass margins, these types of things, creating some of that ecological infrastructure that's particularly in our more poorly producing areas. Let them be a refuge, let them provide habitat for insects who can then migrate into the productive areas and provide some other ecosystem services. So I think practically speaking, those are some of the strategies. And I'll close with this slide here, just to say that returning back to a kind of this opening discussion, the classic kind of view of soil health, chemistry, physics, biology, the three-legged stool. We've got to make sure we optimize these processes, the chemistry, the physics, the biology. Let's change that soil. Let's improve that soil. I think that's all fine. Very good way to think about soil health in the context of that. But arguably with that top-down approach that we've kind of now tried to share some examples of the way in which plants can change the soil, the way in which diverse mixtures of plants can also change the soil, that really our soil health thinking really should look more like this. It should be a 4-part Venn diagram. We should sit the role of plants and how they interact with physics, with biology, with chemistry, and how all of the system interacts. Of course, it is a two-way street, back and forth, as I mentioned, but I think that putting plants and the way their contribution to this discussion, I think, is also a very important piece of the puzzle. So the question was, I've shared a lot of examples about companion planting, root-to-root interactions, these more diverse interactions, and is there any good resources in which you could find more information on this? And it's a good question. I would say I spend a lot of time, as you can see, reading a lot of the literature, and I would say that we often talk about how science and practice is disconnected. What's happening at the farm level is not always being well represented in the research. I would say that one of the examples, thankfully can say this, that seems to be a really strong alignment at the moment is this intercropping example. I'm seeing very good interest and traction at the field level, and there are many, many studies on intercropping happening at the moment. There's some really, really good. I only skimmed a few of them. There is a lot of really good information happening in that space at the moment. So I'm spending a lot more of my time there, trying to get some of the latest stuff, and that is definitely a good resource, but I know not all farmers want to wade through scientific literature, of course. And so beyond that, I would say that the usual avenues, I think that most of what's most interesting happening in this space is from other farmers. So things like the social media channels, Twitter, following farmers who are doing this and learning from them, I think is probably one of the best the resources at the moment. And again, you can find the right hashtags and search for intercropping on Twitter and whatnot to try and find some of these examples from all around the world, which I think is also very useful. But yeah, on some of the companion things, companion planting, of course, is traditionally used much more in the gardening world, and you could argue that some of the old gardening resources would also be maybe a useful role. But otherwise, to be honest, I think the best, if you just do a bit of Googling on this, you will come across some resources, and I would encourage you to just use things like Twitter to see what other farmers are actually doing.